Hello, this is an educational video produced within the Edgenuity family. Students of mine asked for some additional help in order to improve their score on a test. And so this is a kind of a collection of information that they are struggling with. Struggling is not the right word, but needed help with. And so the purpose of this video is to help them to improve their score on a test. It's the macromolecule test. And the purpose is not to just tell them the answer to select. The purpose is to kind of fill in their information gaps on a variety of topics which do have a common theme, luckily. That common theme is the movement of energy through the system, which encompasses the photosynthetic process and the cellular respiration process, and how that energy is then can be used. And of course, as you can see by what's on the screen, the analysis and best understanding of diagrams that showcase energy. And so that is the purpose. So if you are not the person that this video is intended to, I hope it also helps you if you are looking for this kind of material. And so with that, we will get started. Some of the topics that we're going to be talking about, obviously, as you can see, energy reactions, enzymes and the work that they do. We will also get into macromolecules and their monomers, and how to take those information and to analyze questions and be able to answer them in the appropriate manner. So we'll begin here at the top. This is 5 questions that they took the practice quiz a couple of times, and of the 5, 3 of them they missed both times, which really emphasizes the need for the material. And so we will walk through them again, not with the purpose of pointing to the answer, but filling in information gaps so they can analyze on their own and determine the right answer. So we're going to start here, and let me grab a pen, and let's go with a nice teacher red here. We're going to start here first in this section with the enzymes. An enzyme is a protein. Proteins have many functions, and their structure varies greatly. But enzymes are one of those. And the job of an enzyme is to take a substrate, as you can see here. Let me circle that right there. Take a substrate, and it attaches to the active site, which you can see there, and it fits very nicely in there. And it's very much like a lock and key type system. The enzymes are designed for those substrates, and if the substrate doesn't fit in that section, then the enzyme cannot work on it. And there are some things that can alter that or hinder that, as you can see in this diagram. But that's not the purpose of that information, not the purpose of this video. But it's the idea that the substrate is what attaches and then also is released from the protein. It may alter it if that's what the function of the enzyme is to, but the idea, the substrate attaches, the work gets done, and then the substrate releases from the enzyme. So the idea, when you're talking about components, we talked about active site, and of course all of this is a chemical reaction in the enzyme, which is the understanding of that process is why it's really important that you have a good solid chemistry foundation in order to understand what's going on there. So hopefully these diagrams and this information helps to answer that particular question. Oh, didn't mean to grab onto that. Moving this over just a little bit, and now we will look at this question here. The question is, which statement best describes the relationship between an activation energy and the rate of reaction? And it's a multiple choice question, as is the enzyme question that we previously worked on or looked at. It's a multiple choice question. And again, we want to provide information as a way to help analyze and to understand. So we're looking at here the relationship between activation energy and the rate of reaction. And this will also pair into nicely the next question that we will be looking at. But here's the idea, and we're talking in this work here, the reason why I put these side by side is this work that's being done here is being done by an enzyme. Okay, enzymes help with this. Enzymes are super important. We couldn't digest our food without an enzyme. And actually, we would starve to death without the help of enzymes and things that speed up the reaction time of breaking down our food. Our body just wouldn't do it quick enough. But this idea, we're going to look at this purple line here first. This purple line represents the process, both in time and energy, that the reaction would need to go through. And you can see that the reaction would actually happen here. Okay, and that's the energy that is needed to activate the reaction. And when we say reaction, you could think, as far as energy, you could think heat, you could think whatever gets it going, but usually it's energy. And so, for the reaction to occur, if it's got to reach this level of energy, and the y-axis here doesn't have any units, so we don't know what level that is, but it doesn't matter. It's the relationship we're talking about. It has to reach this, and when it does, boom, the reaction will happen. But what an enzyme does as far as the reaction rate, or activation energy, which is what we're looking for here, it simply lowers. And that comes down when an enzyme comes in and provides that activation energy, it lowers the energy that is needed to start or initiate the reaction. In other words, it pushes it down. Okay, we'll draw this big arrow down here. It squeezes it down. A metaphorical example would be in the town that I work in, there's a very large hill by our middle school, Atlantic Street. It's a really tall hill, and to ride a bike up that hill is extremely challenging and difficult. It can be done, especially if you are a middle schooler headed to school, but it's very challenging. So if you can imagine, an enzyme would come in, and at the peak of that hill, it would just push it down to make it easier for the biker to ride up that hill. That's what an enzyme does. It lowers the activation energy. So if the relationship between activation energy and the rate of reaction, when it's lowered, then the reaction can happen quicker. Okay, so that's the relationship between activation energy and rate of reaction. When the activation energy is lowered, the reaction can happen quicker. Okay, so hopefully that helps to answer that question. And I'm going to close my ink tray here and roll this up. No, I don't mean to do all that. Roll the whole thing up here. There we go. And that leads us into this graph. This graph is a very nice graph. It has multiple components, as most graphs would. And let's talk about what this stuff means. The yellow line is an exploding firework, and the blue line is a burning candle. And if we're thinking activation energy, you can see at this point here, let me get my pen back up. At this point right here is when the candle would start to burn if we're thinking activation energy. And at this point here, okay, the firework, the gunpowder and everything in there would begin to burn. But we've got to do some energy, get some energy going to get that started. So if we're burning a candle, you can imagine lighting a match, or in these more modern times, a little aim flame. Click that, push that trigger and get that flame going. And once that wick is lit, then that reaction can begin to happen. And here, if you can imagine, 4th of July, you got those, look at those punks going, it's lit, that cherry red end, and you put that up to the, just lost my word for the, it's not a wick, what's the word? I don't know, but you know what I'm talking about. light that, and then that burns through, and boom, the firework begins to explode. Okay, so that would, if you can imagine, this is an amount of energy. Okay, and now here we've got time, which is a fantastic variable. So if this is the position, okay, so these are going to happen at the same time, as far as time and for the seconds or parts of a second. But when you look at the reaction itself, okay, and because this ends at this slot and the candle ends at this slot, if you think of time, then this would be a quicker time. If we did this in seconds, maybe say this is 1 second, and 2 seconds, and 3 seconds, and 4 seconds, and 5 seconds, then the exploding firework would happen in 4 seconds as opposed to candle in 5 seconds, which probably isn't accurate. That's a candle would take longer than that. But you can see that the firework would happen quicker. Okay, quicker. And when we think about the amount of energy, total energy, looks like the firework, because it's underneath the blue line, would have less. Okay, it peaks right here, and obviously the candle peaks right there. So that amount of energy would be less than the firework would have less. But so we're talking quicker. That's the main thing we want to think about, is quicker. So the question is, the graph below compares the rate of reaction of a burning candle and an exploding firework. So we're talking about time. Okay, when we say rate of reaction, okay, we're talking time. And so the firework would happen quicker. Okay, quicker than the candle. Okay, so let's close this and move this down to the last two topics. And if we get our pen back up, you can see that we're talking about macromolecules here. Okay, so let's analyze the question. The phrase energy storage. Okay, if you're talking energy storage, you're talking the processing of energy. So you're talking cellular respiration. The cell has broken down the glucose molecule that the plants made during photosynthesis, and then has now broken it down through cellular respiration, and your cells are ready to store it as energy. And when that happens, that molecule is called glycogen. In an animal, which as humans would be part of that. If it was a plant, and you can see this here, if it was a plant storing that same sugar molecule, that carbohydrate, it would be as starch. And if you are familiar with at all the diets of athletes, starch is a major part of that, to get that energy, to store that energy so it can be used at the appropriate time. So we're talking glycogen is and here the word is here, storage, energy storage in animals, which we fit into that category, which is stated in the question. Okay, so and the next part of the question is, a hormone that is called insulin controls the storage of glycogen, okay, in the liver. Insulin is made of amino acids. Okay, and so the question asked, which statement correctly identifies the type of macromolecules that are described? In other words, which types of macromolecules have they been talking about? And again, it's a multiple choice question, but again, we don't want to just to point to answers. We want to fill in the information gaps and help students to analyze and come up with a logical conclusion, and then be able to answer the question on their own. So if the insulin is made of amino acids are monomers. In other words, they are simple molecules, individual molecules, okay, that make up something larger. When they are monomers, they are individual, and when they're combined together and making a molecule, that would be a polymer, okay, a long strand or a long string. Amino acids are the monomers, okay, as you can see right here, of proteins, okay, which means if amino acids are the monomers of proteins, okay, and insulin is made of amino acids, then that means that insulins, okay, are proteins. It's just important to understand that connection between the monomer and the final product. So insulin would be a polymer, okay, and while the amino acids are monomer. Mono meaning one, poly meaning many, and that's the idea behind this question and the connection with the macromolecules. Okay, and closing that, so I can move this up for our final topic. Okay, and get my pen back. Okay, the question asked, and this is the question here. Again, it's multiple choice, and we're going to try to fill in that information gap to help with the question. Which macromolecule forms a double layer as a primary structure of the cell membrane? Okay, so we have to understand the components of the cell membrane, and there are many, and it's a fascinating part of your cell that I really enjoy talking about. But some of the things that are in here, we got these proteins. Okay, these proteins are part of the process of getting materials in and out of the cell. We've got the glycolipids that are part of the recognition, glycoprotein. Again, part of the recognition process that molecules have to go through to be recognized. Much like if you arrive at a military base or a large distribution center, you meet the person at the front guard gate, and you got to announce who you are, and they got to check their list to make sure you're expected. And if you're expected, you're allowed to go in. If you're not expected, then there may be some due diligence to verify your need. And if you're recognized as a needed item, then you're allowed to come in. And if you're not, you are turned away. That's what the protein's job is to do. But what we want to focus on is these components right here. That's a phospholipid. Okay, a phospholipid is what makes up the structure of a cell. It forms this bilayer, okay, or a double layer. In other words, part of the phospholipids are facing to the outside, and part of the phospholipids are facing the inside of the cell, and they form that through the processes. There's a lot of things at play, but to make it simple, with the idea of their polar heads that are hydrophilic and the tails that are hydrophobic in that process. So that is the idea behind the double layer and the plasma in the cell membrane and the components within. So I hope this helps. I hope this video helps you to achieve your goals on your macromolecule test. And with that, we will say goodbye.